So in the enzyme lab, most people had basically the right setup for their graph. They identified pH as our independent variable, the thing that we were testing. So what went on the x-axis, and that was the cause that would affect the enzyme reaction rate over here on the y-axis. And then they had a title, pretty minimal title. I think we could do a bit better though. So let's create a little more detailed title, including something about the pH and enzyme activity, but going a bit further than that. There we go. How about that? So we got all the elements that tell you a lot about what we did in the lab. That it was hydrogen peroxide converted to oxygen. The enzyme, that's a pretty important part. Even that we got it from the potato, that it was in water and tested different pH levels. All right, so that's a good descriptive title. We could even have added something about this visual estimate method, but we'll leave it at that. So the next thing that I saw some errors was on graphing the pH and setting up the x-axis. So we tested pHs of 4, 6, and 9. But you notice how here there's a jump of 2, and here there's a jump of 3, but they're spaced the same. So that's not the way we want to do that. Let's try this instead. So we'll erase that 9, put 8, 10, 12. So we've set up our graph already. Might even add 2 over here. Nice and evenly spaced out so it doesn't get all skewed. Okay, so let's go to plotting the data. Most people didn't have much of a problem with this one. We had a moderate amount of activity at pH of 4, a little more at 6, and most people had the most at 9. Notice 9, I'm plotting kind of right above that. So we had to interpolate a little bit, but no problem. Then we want to draw a trend line. Some people added even a little label in here noting that this was vinegar and this was water and so forth. But I think with that, we have a really good graph, tells us what the results of the experiment were. Let me also add something else. What can we generalize from this? Let's get rid of the old data and let's ignore catalase. Let's just kind of ignore this whole thing and say, if we're just talking about any enzyme, then it's most likely to have an optimal pH of 7. So the highest activity, the optima, is going to be around 7. Again, this is just for most of them. As we move toward the acid side, then the reaction rate will go down. And as we move toward the basic side, the reaction rate will also go down. Why is that? Well, because remember, enzymes have that active site, and as the pH changes, then that active site and the whole enzyme in particular is kind of crumpled up and destroyed. It becomes denatured. And so as it loses its shape, it loses its function. It's kind of slowly. So what I would expect to see is a curve a little more like this, where the peak is at 7, and then it goes down to nothing as we get into really acidic or really basic. Some enzymes are even more sensitive, so you might get this really steep curve like this, where it only works in this pretty narrow range from about 6 to 9. Catalase worked a little better at a pH of 9, it looked like. What about other enzymes? Well, I'm glad you asked. What about something like pepsin? Pepsin is an enzyme that works in the stomach. The stomach is a pH of about 2, and so I would expect to see a curve kind of like this for pepsin. So not all enzymes have the same optimal pH, but most of them kind of follow this purple curve, work best at a neutral pH. What about it for temperature? Let's explore that for a moment. As for the catalase lab, we tested these temperatures, but again, we want to have our number line, our x-axis, drawn out proportionally, so equal spacing between 0, 50, and 100. So we tested about 8, and so that had a fairly low activity. 17 was just a little bit more than that. It had a higher activity. 40, that's about in here, had the highest activity, but by the time we got to 69, then we were at basically nothing. So notice how this is spread out, that there's a little more space between these two than there is these two, and a lot more space over here. So this gives us a truer sense of how these enzymes react at different temperatures. So when we draw this trend line, it's actually curved, and we could then estimate, for example, what if we want to know how would this react at 50 degrees? We would estimate that the enzyme activity is somewhere right around here, kind of moderate. So that gives us the ability to do so. So that's part of the reason we want that trend line to be, or the axis to be proportional and those numbers spread out that way. Well, what about a more general curve? Let's erase some of this stuff and look at that. Let's generalize. Let's not talk about catalase, but just general for enzymes. Enzymes found in the human body. Well, we don't operate at 0, that's freezing. 100 is boiling water, and 50 would be a really, really, really hot day. We're here around 37 degrees Celsius. That's our normal body temperature. So I'd expect the optimal pH, so the highest activity would be around there. To the left of that, as we get colder, everything just moves slower. And so reaction speeds tend to slow down as we get colder and colder. And then once we get past 37 degrees, we start slowing down, but for a different reason. And what happens there is we start to slow down as that enzyme starts to get shaken around and unfold. And then usually right around 60 degrees is about the maximum tolerance for most enzymes. And after that, the activity rate is just 0. And so this is due to denaturing. Again, kind of like in the pH, that enzyme requires to be a specific shape for the active site. But when you get really hot, the activity in here just increases. You're getting all these molecules just bombarding this thing and just kind of cutting it to shreds in a way, shaking it apart. And so the active site doesn't work. And we would see a trend like this with the optimum, again, around 37 degrees, somewhere around there. It's going to be different for different organisms. So something like archaea, those guys that live in thermal hot springs, then I would expect to see a curve where that optimal is way out here, maybe even over 100 degrees. But that's pretty extreme. Most of ours is going to be more in the kind of moderate temperature range.